all of our advances now, the median progression-free survival is so long, do you think the FDA, or what data do you think the FDA need to be to be able to think about this as an endpoint?
I think with our success, we have a problem, which is what you are alluding to, that PFS is so long that comparing drug efficacy takes years if we still follow PFS. And I think everybody, including FDA, is on board that we need some surrogate to have a shorter time to evaluate the response. And so there has been really significant effort ongoing to collect all the data we have, all these 7,000 to 10,000 patients, and then analyze it very stringently, scientifically, to confirm the surrogacy of MRD as a reflective of PFS, which reflects OS. The ongoing effort where all of us have gotten together, sharing data, and what FDA needs is a patient-level data. So I cannot use your published study and my published study and Mary's published study and come up with a meta-analysis, which we all done. We need patient-level data and data from studies which are randomized study. So you show a benefit of MRD on both the arms, which is one of the critical things they need. Fortunately, we have these studies. We almost around 10 to 12 of such studies, including relapsed disease and newly diagnosed, old patients and younger or transplant eligible. And so now it's time to analyze it. And once we have that, I think I'm very hopeful that we may be able to provide enough material to FDA for them to accept MRD as surrogate for PFS. And then our life and drug approval process might become much easier or quicker.
Yeah, I was going to say, Maria-V and I were talking before we came on air about how the ideal test would be agnostic of the method and of the treatment. So it didn't matter how you measured the MRD as long as you were to a level, and it didn't matter how you got to being MRD negative, you had the same kind of clinical outcome.
I think that's exactly what, when we had a meeting with FDA a few months ago, and that's exactly what their point was. So they are agnostic and we are agnostic to method. I think Mary made a very important point. If done well, both tests are equal, but done well is a key word. So it doesn't matter how you do it. Your depth should be 10 to minus 5. I think 6 is better, but 5 is more routinely achieved by most of the tests, and so that may end up being our sensitivity, and we'll have to see what FDA accepts. But I think keeping that in mind, doesn't matter what treatment got you MRD negativity, or at what time point, although time point may become important factor in all this analysis. But keeping all this in mind, it's the end point that is important. One thing I would really highlight for us in practice is that normally the first bone marrow we do, that goes to the pathologist. The second goes to patient cytogenetics, and third then goes to somewhere. The fourth bone marrow goes for MRD. And this may be important at the diagnosis time. However, when we are doing subsequent measurement for the purpose of MRD, I think we have to all make in our institution and in general a pitch that the first bone marrow should go for MRD measurement, because otherwise we get a little bit dilute marrow. And again, I will say one more thing before you ask, is that, so for flow measure method can measure some dilution effect, the sequencing doesn't. But when we go to the depth of 10 to minus 6, even if the marrow is diluted 50%, which is usually the maximum, we are decreasing our sensitivity from 10 to minus 6 to 5 times 10 to minus 5. So the dilution is not as bad. Even if it's a 90% diluted, it's only 1 log. So 10 to minus 6 sensitivity becomes 10 to minus 5. So I'm not too worried about dilution, but I think the first marrow going still is an important factor that we should all keep in mind and push our pathologists and other to let us do that.
So just in closing, there was a bit of a curveball that got put into the discussions this morning, because the Cassiopeia data was presented by the French group, and they'd compared the NGS data to the next generation flow data. And there seemed to be a little bit of discrepancies between the two. Do you have any thoughts or comments?
Well, Hervé did just the opposite well, just the contrary what you said. The concordance was quite high, approximately over 80% for next generation flow and next generation sequencing. I think that the main problem from my point of view for this comparison is that, they put 10 to the minus 5 for flow, 10 to the minus 5 for next generation sequencing, and maybe they reach 10 to the minus 6 to next generation sequencing. So the comparison, I don't know if it is completely appropriate. Point number one. And the point number two is when you look at the curves for progression-free survival, for me it was a bit intriguing why minimal residual disease negativity was not agnostic for the treatment arm, because patients receiving DVTD were better than VTD alone. And this is, from my point of view, this is not correct, because according to our discussion, both arms, when MRD is negative, have to be on the same level. So I don't know. The other point to discuss about the Cassiopeia is the discrepancy between patients achieving stringent complete response and minimal residual disease negative, because it's almost the double of patients achieving MRD negativity. It's true that they selected the stringent complete response, but for me it's a bit of discrepancy in spite of they decided to use the intent to treat patient population is correct, but there are some caveats from my point of view.
Yeah, I think I totally agree. That's the right analysis. I think the comparison between the two methods was, I think there is something more to that.
And just you have to go, for example, for the French study, they did with VRD transplant versus no transplant and maintenance. And the MRD evaluation pre-maintenance was able to select as a group of patients with an excellent outcome. You go to the Spanish trial, VRD transplant, consolidation, maintenance, MRD next generation flow before maintenance. Patients with MRD negative, similar outcome to the French group. This is what you can compare, and the outcome is excellent for patients using different methods, different treatments, not exactly the same, but the outcome is exactly the same.
I think same thing is true for Castor and Pollux.
Yeah.
The both arms are very overlapping if they're MRD negative.
Yeah.
So it's the same point.
No, I usually say that if you give water to your patient and MRD is negative, the outcome is going to be excellent.
Same thing. That's a good point.
Excellent. So thank you so much for the discussion. I think, as you say, we've been agreeing on many things now in MRD. We've moved from so much disagreement, we now have a lot of agreement. And moving forward, we just need to, I guess, polish and make sure that we're asking the right questions and providing the right answers to the FDA so we can move forward with it. So
Absolutely.
Thank you very much for joining me.
Thank
and
you.
Thank you everybody for joining us, and we'll see you soon from the International Myeloma Workshop. Thank you.